Well, welcome back to our lesson. And we were talking about looking at the details of each organelle structure and function. I just want to say something encouraging and maybe a bit of a warning that this part is usually there's a lot of information and you might feel very overloaded. And it might be that you feel a bit dry as well. But don't worry, it took me a while as well when I was a student to get my head around each and every organelle. But I think look at it a bit like, going into a museum and into the cell and seeing, different parts of the cell that they do different things. Actually, after a while, it can be quite interesting. So let's start out with the most obvious organelle in a cell, the one that's found right in the center, the nucleus. So a nucleus, you can actually break it down into more parts. A nucleus, the structure, it has a nuclear envelope. Okay, it's its own membrane that surrounds the nucleus. And this nuclear envelope surrounds the nucleoplasm, a substance inside the nucleus. Not cytoplasm, but the nucleoplasm. And right in the center of the nucleus, you'll find usually a very dense region, very densely stained, called the nucleolus. And finally, inside the nucleus, you also find thread-like chromatin, which I'll talk a bit more on the next slide. So this chromatin, these threads, you see all these threads in the nucleus? Well, they are actually made out of DNA and protein. So DNA associated with protein gives us chromatin. And this DNA is very important. DNA, you might have heard, contains genetic information or hereditary information that we get from our mother and our father and that we pass on to our children. And it also contains instructions for cell activities. For example, cells can make many different types of proteins. And it is the DNA that contains instructions for how to make these proteins. The DNA is a bit like a blueprint, and without the DNA, the cell is not able to do much or carry out living processes on its own. Now, it's worth saying that this chromatin doesn't always look like a thread. During cell division, that means when the cell divides, it can actually condense to form what you can see visible structures called chromosomes, which look very cool, like X, Y structures. And we'll talk a bit more about this in our topic on cell division. Now, because the nucleus contains the instructions in DNA, its main function is to control cell activities. And it also controls cell division. It's like the control freak of the cell. It controls, do this, do that. It has all the information in there. Okay, moving on to the cell surface membrane. Now, cell surface membrane is made out of lipids, which are fats, and proteins. It is partially permeable, which means that it allows some substances to pass through and does not allow some to pass through. So we say that it controls the movement of substances in and out of a cell. It is a bit like this. If you've been to the airport, this is the baggage check, right? Yeah, recently when I went overseas, I had a baggage check as well. And the cell membrane is a bit like that. It checks and it allows certain things to come in, but doesn't allow certain substances to come in. And that's really important because you want to maintain the difference between inside the cell and outside the cell. Because some things are needed for life and some things you don't want it in the cell. On to the rough endoplasmic reticulum. This is new for you, and also the ribosomes. How do we describe the RER? We say that they are flattened spaces surrounded by membranes. So you see here, this is the rough endoplasmic reticulum, this area. Okay, you see that they are actually flat spaces surrounded by this membrane. And notice that the membrane is actually continuous with the nuclear envelope. See here? Nuclear envelope becomes the membrane. It's continuous. It's actually connected to the nuclear envelope. if you trace it. And more importantly, they have ribosomes attached. These little dots are the ribosomes. Okay, what does the RER do? Okay, before that, okay, so you can see, right, this drawing 2D. Okay, this is the nuclear envelope here, this blue color part, nuclear envelope. And you can see that the rough endoplasmic reticulum is continuous. That means it's kind of like connected to the nuclear envelope, it's part of it, right? And the little red dots are the ribosomes. They are bound, attached to the rough endoplasmic reticulum. Now what does it do? The ribosomes themselves, well, remember they use that, say the nucleus had very important information. Well, the ribosomes use that information to synthesize proteins. Can you try to learn this word synthesize? Right, you're not in Sec 1 anymore, so try, it means make. But scientists, the word synthesize is a lot more scientific, huh? So ribosomes use information from nucleus to synthesize proteins using amino acids. So they basically join amino acids together to form proteins. So this is a ribosome here, and you can see that it uses the instructions from the nucleus. The instructions is like this blue line here, right? And the ribosomes, what they do is they actually move. They move along the instructions. They read it a bit like a barcode scanning. And as they scan, they connect the amino acids together to form a protein. Now remember I said that there's some ribosomes that are attached to the rough endoplasmic reticulum. Now this is a bit extra information, but the attached ribosomes tend to make proteins that are transported out of the cell or secreted, that leave the cell. While the free ribosomes found in the cytoplasm, they tend to make proteins that are used in the cell. So there's just a bit of a difference in the kind of proteins that different ribosomes make. So then what does the RER itself do? Well, the rough endoplasmic reticulum transports proteins made by ribosomes to the Golgi apparatus. Okay, so once the proteins are made by the ribosomes, they are packaged into vesicles. You see here, this is the rough endoplasmic reticulum here, the diagram at the bottom, and you see a vesicle that is budding off. The vesicle is just like a membrane. The purple things are the proteins. So the proteins are packaged into these membrane-bound vesicles, and these vesicles move to the Golgi apparatus, where, as we will see later, something happens to the proteins when they reach the Golgi apparatus. So that's the RER. Now, closely related to the RER is the smooth endoplasmic reticulum. These are also flattened space. These are also spaces that are surrounded by membranes here. Can you see the smooth endoplasmic reticulum surrounded by membranes? But they are more tubular. That means like tubes, okay, not so much flattened. So not so much flattened, but they are more like tubes. And importantly, very importantly, they have no ribosomes. See, very smooth and nice, like the smooth skin that a lot of girls want. Now, what does the smooth endoplasmic reticulum do? Well, it synthesizes, that big word again, please learn it. It synthesizes fats and steroids. Steroids are not the kind of thing that those muscle people think to go very big. Okay, steroids are a kind of hormone that you find in your body. So like for girls, for example, you might have heard of estrogen, that's a steroid. That is the smooth endoplasmic reticulum that makes that. The smooth endoplasmic reticulum also does detoxification of harmful substances. Okay, like alcohol. If you drink a lot of alcohol, the smooth endoplasmic reticulum has to work very hard to break it down into something that is not harmful. So looking at these diagrams here, you see that under the microscope, the electron microscope, the smooth endoplasmic reticulum usually looks like these tubes, right? You can see that they're very tubular, and the rough endoplasmic reticulum is more flattened, like that. Okay, you can see the 3D image here. And that brings me to the Golgi. So this is the stacks of membrane. Okay, again, stacks of flattened membrane spaces that are usually a bit further from the nucleus. And remember the vesicles from the ER? Remember those vesicles containing your proteins? They will move towards the Golgi and they will actually fuse with one side of the Golgi apparatus. Fuse for what, right? Well, the Golgi apparatus is a bit like a post office or warehouse. Okay, what it does is that it will modify substances made by the endoplasmic reticulum. For example, it can modify the proteins that are made slightly. Okay, just modify it slightly so that it becomes better or more useful. And then what it does is that it will then package these modified substances into another vesicle. And most importantly, it will sort the vesicles to go to different destinations. Okay, so a bit like a post office, it will say, okay, let's take in the substance, let's modify it a bit, and then let's send it out to the final destination. For example, the cell membrane. Or it could be going to a different part of the cell. Or it could be going to, back to the nucleus. Well, it is the Golgi apparatus that decides where things go. So let me bring it all together before we end this section and this little video. How are proteins made? Okay, a very common thing that they might ask you is how are proteins made? And let's do this to summarize as well the function. Well, remember that the instructions are contained in the nucleus in the form of my chromatin, right, my DNA. And these instructions are used by the RER, in particular the ribosomes, to make and to transport proteins to the Golgi. See, we see the vesicles that pinch off and move to the Golgi. The Golgi apparatus modifies and packages the vesicles and sorts them out to other destinations. So for example, it could be going to the cell membrane here or to other destinations here. And when the vesicles that go to the cell membrane reach there, okay, they fuse with the cell membrane and the proteins are actually secreted or released into the environment. So these four organelles, the nucleus, the ER, Golgi, and the cell membrane, they actually work very closely together in making and sending out proteins to the environment. And with that, I just want to conclude this section, so just take time to read.